Okay, so this is lab video 5, and I'm going to cover the BAP, the nitrate, Snyder, and Kirby Bauer in this video. So the blood agar plate is not, you're not going to be doing a P&P for this assignment, but you may see some questions on the exam, practical exam, about it. The important thing to remember for that purpose is that red blood cells are in the BAP plate. They act as an inhibitor and an indicator. So they inhibit some bacteria. And they can produce, the red blood cells can produce 3 types of hemolysis. If they grow and don't affect the red blood cells, you say it's gamma, no hemolysis. There's actually a gamma sign here. The bacteria you don't see a heavy bacteria growth, unfortunately, in this image. The beta sign, you can clearly see that. That's where the bacteria grew. They just drew a beta sign with their loop. And they completely hemolyze the red blood cells. So you got beta. Then there's an alpha sign. And alpha hemolysis is in between the gamma and the beta, right? It's not complete, but there is some hemolysis. So they call that incomplete hemolysis. So just know the definition of these 3 types of hemolysis on this plate. Now nitrate is this one is a little bit complicated. So, right, in the purpose, nitrate is used as the final electron acceptor. If they're using it as the final electron acceptor, then they're using anaerobic respiration as opposed to aerobic. Okay, they're using nitrate as the final electron acceptor. This is nitrate, and it goes to nitrite if they do. So the test is pretty straightforward. You take this tube, they grow in it. You add nitrate E and nitrate A and nitrate B. If you get a red color, right, it's detecting nitrite. So reduction did occur. Now if you don't get a color, then no change indicates one of the following, then you have to do a confirmation test. So then your principal should include the following, right? Nitrate, it could be that nitrate wasn't reduced, or it was. It could have been what's called further reduced, right? If it wasn't reduced, you wouldn't get a red color because you have you still have nitrate there, or you might not have a red color because nitrite, this guy, was further reduced. So you have to decide, well, that's the case. So what you do is you add what's called zinc powder or nitrate C. What zinc does is converts if there's any of this left, nitrate, then it converts it to nitrite, so you get a red color after you add zinc. So that confirms the negative result. But if you add zinc and you don't get a red color, then you further reduced it from nitrite, right? So all of that is listed and the explanation is given on the PowerPoint, right? It's complicated. Don't worry about how complicated it is, right? You might, on the practical, I might expect you, if I added nitrate A and nitrate B and got a color, what does that tell you? For instance, if I added A, B, and C, and you get a red color, that means it's negative, and you need to tell me that's a negative test. So if you remember the scenarios, what occurred, you shouldn't have any difficulty with it. This determines if they're using anaerobic respiration. Now the Snyder media, or agar, that's the name of this tube. It's used, the purpose of it is to detect lactobacillus in saliva. Right, the indicator is brome cresol green. It starts out as a green color. If the bacteria you add, like lactobacillus, are present in your saliva, that's what you'll be adding to the media. They'll break down the glucose, and the brome cresol green will turn yellow. Okay. Now, if we did this experiment, the media starts off as in a liquid form, right? We keep it warm enough so it doesn't solidify. Then you add your saliva, you mix it up, and then it turns to solid pretty quickly, and the bacteria will grow. And this isn't something, lactobacillus or dental caries is how bacteria that can generate cavities, but that doesn't mean dentists use this ever to determine if you have cavities. So it's just a fun experiment to do in lab, if we were doing the lab. But unfortunately, you're having to watch this video of me explaining it. So the last experiment for this lab would have been the Kirby-Bauer method. And the Kirby-Bauer method is used to determine antibiotic sensitivity / resistance, right? If bacteria are resistant to antibiotics, then you don't want to treat that person with that antibiotic. So this is a way of determining if they're resistant to penicillin or ampicillin. Okay. And the way you do that, right, the principle is the Mueller-Hinton plate is what's used. It's very similar to a TSA plate. And then what you do is you apply an even coating of bacteria to the surface of the plate. You want the bacteria to grow. If we hadn't added any discs to this plate, for instance, you'd have an even lawn of bacteria growing on the surface. Before they grow, though, you add discs that contain, on this plate, every disc contains a different antibiotic. And you can see in this disc, it's a sulfonamide, I think. It has a yellow color. So you can see how it goes from the disc and penetrates the media. All right? And then the further you get away from the disc, the less of the antibiotic or antimicrobial gets into the media, so it's less effective, right? But it's most effective nearest the disc. So you release it, and if you generate a zone of inhibition, then the bacteria are sensitive. That means the antibiotic is effective against, right? If they don't have a zone, like these two discs, then the antibiotic had no effect on the bacteria, and you would consider them resistant. So it's a pretty straightforward kind of explanation, I think. What causes the zone of inhibition? Right, the antibiotic does. Okay. And I think there's also a, I don't know if I removed this or not. Don't just ignore this on the PowerPoint. It won't be on the practical. So that way, I think now the next video I make won't be covering anything on the PowerPoint, and there really won't be any questions on the practical from that, of those experiments, right? We're going to do on the microbiology of food. There's one on blood typing, I think, and I can't remember the other. But that'll be a really quick video probably, and there won't be any quiz questions from it, so don't worry about that.